Hello, today we will be talking about nucleotides, so let's start the video.
Alright guys, today we'll be talking about nucleotides. It's our first video from the USMLE Step 1 series. We'll be covering biochemistry first. So here, we're going to be talking about nucleotides. Here, I have laid down some key points that we will be discussing in our short video. So we're going to be talking about the type of sugar that is involved in the synthesis of nucleotides, then what is their structure about. Then we're going to be comparing the bases, the nitrogenous bases, purines and pyrimidines. Then how each individual nucleotide participates in the formation of a nucleotide chain. And lastly, we're going to be talking about the deamination reactions and what's the theory behind it, what's the science behind it. We'll be dealing with the nucleotide structures so that we can understand them better, so that when we'll be talking about catabolism of nucleotides, so it will be a piece of cake for us. All right, guys, so we'll be talking about structures of sugars that are involved in the synthesis of nucleotides. So we have two types of nucleotides. We have nucleotides which are made up of, nucleotides which make up RNA. We have some nucleotides which make up RNA. And we have some nucleotides which make up DNA. So both are actually, macromolecules, but both have, the monomer or the, basic subunit as nucleotide. So it is also made up of nucleotide. It is also made up of nucleotide. What differs essentially in both of them is the type of sugar that we use in the synthesis of nucleotides. So here, both of the nucleotides differ in the sugar composition. In RNA, we specifically talk about ribose. And when I'm talking about DNA, I'm more concerned with deoxyribose. Now, if I talk about ribose, ribose is a 5-carbon sugar. That being said, that it contains 5 carbon atoms within its structure. So let's start numbering them. So I'll begin with the extreme right side. This one is C1, C2, C3, C4, and this one over here is C5. Again, I'm going to do the same with the deoxyribose over here, C1, C2, C3, C4, and C5. So as you guys can see, that both structures are quite similar, but they differ when we talk about C2. So here you guys can clearly see that C2 over here has an oxygen or hydroxy group within it. And here we have just the hydrogen atom. We don't have an oxygen attached with that. So when I'm talking about deoxy, I'm talking about removal of oxygen over here. And where I'm talking about carbon number 2. Carbon number 2 lacks oxygen in deoxyribose structure. And when I'm talking about ribose, it will have the usual hydroxyl group attached to it when I'm talking about the C2A. Hopefully you remember the differences. Okay, so how do we get deoxyribose out of this ribose? I'm not going to go into detail of it because it's, it's step in metabolism when we talk about the synthesis of de novo synthesis of pyrimidines over there, especially, or purines. But I'm just giving you a hint so that you guys can better remember the name of the enzyme. Whenever we are converting a ribose nucleotide into a deoxyribose nucleotide. Remember, I'm talking about nucleotide. I'm not talking about sugar. So you guys can clearly see that here we have reduction going on. From your very basic definition of reduction, you guys can recall that reduction is basically removal of oxygen or addition of hydrogen. So clearly here we are removing this oxygen and we are reducing ribose into deoxyribose. So the name of the enzyme that we use over here is nucleotide or ribonucleotide reductase. It's pretty much simple. It's ribonucleotide reductase. So here are just some, review for what its metabolism is gonna look like. The reason I'm just writing out some enzymes over here is that so that we can recall them whenever we need to, whenever we're going to study the metabolism of it. All right, so our next slide essentially is all about the structure of nucleotides, what the nucleotide structure looks like. So as you guys can see that we have a pentose sugar, which can be ribose or it can be deoxyribose, and we have had a look at it when we talked about the previous slide. We were comparing both structures, right? So we know what ribose is like and what deoxyribose is like. This is C2 over here. This is definitely C2 over here. So if it has had an hydroxyl group, if it does have a hydroxyl group, so it's going to be ribose. If it has a hydrogen group, then it's going to be deoxyribose, right? It's something that we have discussed. Okay, then now this is C1 over here, C2, C3, C4, and here we're going to have C5 attached over here. Here's going to be the oxygen. Now with the C5, we have a phosphate group attached to it. So phosphate is essentially attached with what the C5 carbon of the pentose sugar over here. So whenever I'm talking about nucleotides, pentose sugar is going to be their ribose or deoxyribose in nitrogen space at C1 and a phosphate group at C5. Hopefully it's pretty much clear. Okay, so we just have, a look at the structure of nucleotide. And what does it look like? That we're going to have a sugar, either a ribose or a deoxyribose. This is C1, which is going to be attached to a nitrogenous base. This is C4. Then we're going to have C5 over here. This is going to be C4. And this is phosphate group. And the C5 will be connected with what? A phosphate group. That makes up what? A nucleotide. Now we need to differ between the structure of a nucleoside and a nucleotide. You guys can easily see that both have sugar, both have a nitrogenous base. What they don't have, what the nucleoside doesn't have, is a phosphate group. You definitely have a C5 over here. It's not shown in diagram because C5 is the part of what? The sugar. So it's going to have the C5, but it won't have the phosphate group with that. So remember, when I'm talking about nucleoside, it doesn't have the phosphate group. And when we're talking about nucleotide, it has all three essential structures, the nitrogenous base, the sugar, and their phosphate group attached, the C5 carbon. All right, now next I'm going to be talking about the nitrogenous bases. What are they? How many types do we have of them? And how can we differ or differentiate between the structures? Okay, so the first type of nitrogenous base that I'll be talking about over here is going to be pyrimidines. So pyrimidines and purines, how we're going to differentiate between them, that pyrimidines essentially have one wing. So here you guys can see three pyrimidines. We have cytosine, we have thymine, and we have uracil. And all three of them have a one single wing, no double wing structure, as you're going to see in purine. Yeah, over here, we are not seeing any double wing structure. We're going to see that in purine. All pyrimidines have a single wing, as we can see it over here. So how are we going to differentiate between three of them? So I'll begin with, I'm going to begin with cytosine. Okay, I'm going to begin with cytosine. And then I'll move towards, uracil. All right. So here you see cytosine. Cytosine has an NH group over here, an oxygen over here, nitrogen over here, and an amine group over here. So this is essentially what? An amine group. So essentially, if you want to differ between cytosine and uracil, there are other key differences over there as well, as they also differ in the composition of, the N nitrogen over here. But I'm not going to talk about that because it's not something that we can easily remember. So what you're going to differ, or how you can easily tell the difference between cytosine and uracil, is that at the top position, as you can spot an amine group over here, you're going to find an oxygen having a double covalent bond with the carbon over here. That's how you're going to differentiate between that. This is an amine group. Here's an NH2, and here we have an O, which has a double covalent bond with the carbon over here. So this is how we can easily differentiate between cytosine and uracil. All right. Next, I'm going to talk about uracil and thymine. So how are we going to differentiate between uracil and thymine? So here we have a carbon, right? Here we have a carbon. Here the carbon doesn't have a methyl group. But when I'm talking about thymine, it does have a methyl group over here, CH3. So here you're going to find what? A methyl group. All right. Neither cytosine has a methyl group nor uracil has a methyl group. So if I'm talking about thymine, so methyl group is a characteristic finding in the thymine base, something that you won't find in cytosine and uracil. NH2 group is a characteristic finding of cytosine, and that's something you won't find in thymine or uracil either. Uracil is the intermediate base. If I'm talking about the metabolism, so uracil is something that we use as an intermediate from thymine and cytosine. In other words, uracil, when it's formed, is later converted into thymine and cytosine. Hopefully it's clear enough. You can remember these differences, and hopefully you won't be confused in it again. Okay, now next I'm going to talk about another type of base, which is purines. So pyrimidines, as I talked about earlier, that they have a single ring. Here we have two rings. As you guys can see, we have adenine and we have guanine, right? And both have two rings. This ring over here, 1, 2, 3, 4, 5, 6. So we have 6 atoms over here. It is quite similar to what we have just observed with purines. The purines also have what? 6 atoms within its ring, the initial ring. Now this is the first ring that I'm talking about over here. Now the second ring I'm talking about over here, this is the second ring, and it has been derived from imidazole. Imidazole is something which is attached over here in the second cyclic structure within the purines. All right. So remember one thing. Pyrimidines do not have an imidazole ring. Purines do have an imidazole ring. Now, how you're going to differentiate between adenine and guanine is that guanine has a top oxygen attached over here, whereas adenine has an amine or NH2 group attached over here. Okay. So the thing that you have to remember is about adenine and guanine. Here we have an oxygen, and here we have an amine group attached. Now here we can see all the nitrogen bases that we can characteristically find in the nucleotide structure. I've talked about the two of them, pyrimidines and purines, right? So now you have to remember the following points that I'm going to tell you. First, I've talked about two types of nucleotides. One are used in the synthesis of DNA, and the other type is used in the synthesis of RNA. RNA essentially has ribose within its nucleotide, and DNA essentially has deoxyribose in its nucleotide. Aside from their sugar structure, where else do they differ? You guys can clearly see that DNA has adenine, cytosine, guanine, and thymine within its structure. So nitrogen space that we were talking about, it can be either adenine, cytosine, guanine, or thymine. It can never be uracil. All right. But if I'm talking about, let's say RNA. So in RNA, you're gonna find adenine, guanine, uracil, and cytosine. Something like thymine is something that you would never find in the structure of RNA. So that's something you have to remember. Another thing that I want to talk about over here is the complementary base pairing. This is something that I'm gonna review in later slides, but just to give you some information over here is that adenine always binds with either uracil or thymine with two hydrogen bonds, right? This is something that you're gonna remember. So when I'm talking about DNA, so adenine is gonna complementary base pair up with thymine with two hydrogen bonds. And if I'm talking about RNA, adenine will bind with body uracil because RNA doesn't have thymine in its structure. And if I'm talking about guanine, so guanine is gonna form hydrogen bonds with cytosine with three hydrogen bonds. Fine. Now I'm going to talk about the structure of ATP structure, which is adenosine triphosphate. So if I talk about adenosine alone, it is a nucleoside. Nucleoside means that it has a ribose, 5-carbon sugar, and nitrogenous base attached to C1 over here. And this is a C5. And as we're talking about triphosphate, so we have 3 phosphate groups, 1, 2, and 3. And the first destined to covalent bonds or high energy bonds. That means that if we're going to break them, we're going to have a huge amount of energy available to us. Okay, so if I break this bond over here, it becomes ADP, adenosine diphosphate, 2-phosphate group. If I'm going to break this bond over here, then it becomes AMP, which is adenosine monophosphate because mono means 1. So we're going to have adenosine monophosphate over here. So something to remember. All right, now what is the role of ATP? We all know that ATP is the universal energy currency used within the cell. I'm not going to go into the dynamics of it, but just to give you a slight recall, I just mentioned that. The reason I'm talking about the ATP structure over here that I had to name the phosphate groups that I'm talking about over here so that we can recall them later in DNA formation. So the phosphate group, which is closest to the C5 atom over here, it's gonna be our alpha phosphate. All right. And then the middle phosphate group is known as the beta phosphate, and the distant phosphate group is gonna be our gamma phosphate. It's something I want you to remember so that we can later apply this concept to our later slides. Now I'm going to talk about the deamination reaction that we'll be discussing over here. The reason we talked about deamination reaction is that obviously one day you'll be studying about the catabolism of purines or maybe pyrimidines. So we must have enough knowledge that we can differentiate between the structure of cytosine and uracil. Aside from that, we can talk about the resultant product if we remove the amine group in its structure. So deamination essentially means removal of NH2 group. whenever you're removing the amine group or NH2 group, you're basically undergoing a process known as deamination. Now, whenever you're going to be removing NH2 group, you'll be releasing ammonia, NH3 gas. This is something that we'll study in the catabolism of these bases. We're not concerned with them over here, so I won't bother you with that. Now, over here, we have cytosine. And within cytosine structure, you can clearly identify the NH2 group over here, right? So if I'm going through deamination or if I'm deaminating cytosines, obviously NH2 group will be removed and you're going to have an oxygen over here. Remember one thing, that cytosine and uracil differ in one thing, and that is that C4 over here, when I'm talking about uracil, it's gonna have what, an oxygen. And if I'm talking about cytosine, so it will have what, an NH2 group or amine group. So if I'm deaminating cytosine, I'm originally producing what, uracil out of it. Hopefully it's clear. There's no rocket science involved over here. We've already discussed the nucleotide structures before, so it won't be something new. Now talking about adenine. If I'm deaminating adenine, right? If I'm removing the NH2 group over here, so as I told in cytosine, that NH2 group will be replaced by what? Oxygen. So clearly you guys can see that here we have what? An oxygen replacing it. So this is adenine, this is a pyrimidine base, and here we have hypoxanthine, which is again a pyrimidine base. So what differ over here, that adenine has an amine group and hypoxanthine doesn't has an amine group. Instead, it has an oxygen, as you guys can see it over here. That's the differentiating feature between adenine and hypoxanthine. If you're going to remove the NH2 group, you're going to have an hypoxanthine form. When I'm talking about hypoxanthine, why it is important? Because when we're studying about its metabolism or catabolism, essentially, you're going to find out why hypoxanthine is important. I'm not going to talk about it over here. All right, so if I talk about guanine, it's a pyrimidine, right? And here we have xanthine. So if I'm again, here we have NH2 group over here, as you guys can spot it. Now, if I'm deaminating guanine, that essentially means I'm removing NH2 group over here and replacing it with oxygen over here. So obviously, this is guanine. If I move the NH2 group, I'm going to form what? Xanthine. And xanthine again is important. When we're talking about uric acid formation, we'll be discussing xanthine. I want you to remember that guanine, adenine both have NH2 groups. Their position might differ. But when I'm talking about xanthine and hypoxanthine, they have oxygen instead of those NH2 groups. It's something I want you to remember. Here I have one more amazing diagram of different purines, and we're going to be talking about how we can form one. So here we have cytosine, and here I have 5 methyl cytosine. So we can clearly see that cytosine and methyl cytosine means that it doesn't have a methyl group and it has a methyl group. So if I'm undergoing cytosine with methylation, I'm simply adding a CH3 methyl group over here, as you guys can see, right? So when I'm making sure that cytosine is undergoing methylation and forming well 5-methyl cytosine. And if I, like you guys can see that cytosine has an NH2 group, both of them have an NH2 group. So clearly, if I remove this NH2 group over here, with being said that I'm just removing amine group over here with and replacing it with oxygen. So this structure essentially is what? Thymine. Thymine has a methyl group and it doesn't has an amine group. So these are some reactions that I want you to remember, because later when we'll be discussing about catabolism, or when we're talking about their precursors, or when we'll be more concerned with its metabolism, so it will be a lot more easier for us to recall the structure and remember them. Okay, now I'm going to talk about the formation of nucleotide chain. The only reason I was concerned over here that we could identify which carbon groups are attacking which carbon groups, so that it is easier for us to visualize that. Now here I have a nucleotide over here. This is the third carbon attached to the hydroxyl group over here, right? And this is its fifth carbon attached to a phosphate group over here. So this particular nucleotide has a guanine base, and they are classically part of the chain that has been formed earlier, right? Now what's happening over here that there is an incoming nucleotide which wants to be attached or wants to be the part of this chain. So what's going to happen is this third carbon hydroxyl group will attack the alpha phosphate bond. This is the alpha phosphate bond with the beta phosphate bond. When you're going to break this bond, you're going to release huge amount of energy, and that energy will fuel this reaction, and we'll have a phosphodiester linkage being formed between the phosphate over here, the third carbon over here, and the fifth carbon over here. So essentially what happens over here is that this chain is running, and this is again the third carbon over here. We have 5, this N is from 5, is going from 5th to 3rd N, right? So this chain direction is going to be what? 5 to 3 direction. And if I'm talking about this phosphodiester bond, so here I have a 3 to 5 phosphodiester linkage being formed. Phosphodiester. So phosphate simply means that this phosphate group is linking both the previous nucleotide and the subsequent nucleotide. Diester being said that this is an ester bond, this is also an ester bond. So we have two ester bonds, one over here and the second one over here. So this whole linkage is going to be known as phosphodiester linkage or phosphodiester.
Thank you for watching this video. Make sure you like, subscribe and comment any feedback that you may have. Have a nice day ahead.